So, to sum, we can kill these blue cells by targeting apoptosis just to those cells, and we get testes where the hub and the stem cells, germline stem cells, a few of them are left, but if we reverse these conditions, everything returns. And to figure out where the new somatic cells come from, we needed to look at this a little more closely. And so we repeated this experiment, but now we are going to use some markers and characterize the somatic cells that are left, and that happens to be the hub. Now I told you that the hub cells are quiescent, and quiescent means that a cell is reversibly withdrawn from the cell cycle. If you hear about a senescent cell, that's a cell that's thought to be irreversibly withdrawn from the cell cycle. And I'll have to tell you that most of our cells are in one of these two states. There's quite a bit of debate as to how do you distinguish quiescent versus senescent cells. It's actually not that easy to understand, and there are very few examples where this is known, of how the regulation of this is understood in a real tissue. It's a lot better to study this in cell culture because you can have lots of cells and look for markers, but when you get into a real tissue, this turns into a tricky question, and there's a lot of debate about that. So, we began by looking for signs of cell proliferation in these testes that we had done the ablation in, because we're getting a whole bunch of cells, something must be dividing. We're going from very few cells to a pile of cells. So this is a marker here now, so you're familiar with DAPI in blue for all the nuclei. This is the hub. We've now stained it with a marker for an adhesion protein that's really enriched on hub cells. And then this right here is red because it happens to be in mitosis, so it's lighting up with a mitotic phosphoprotein right here. And we never normally see a hub cell dividing, even though we've looked at thousands and thousands of testes. But this is a testis where we saw something we had never seen before. This is clearly a cell inside the hub, and it's clearly in mitosis. So we got very excited when we saw that. And this is all just remaining germ cells that are sitting there. So we looked at this for several days, and on the first day we don't see them dividing. They're still quiescent. But the next day we see a change, and about 12% of our testes start to have this phenomena where we catch hub cells in mitosis, and then it happens for up to about 6 days, but it declines and then it stops. And all the while that this is going on, of course, the tissue is refilling with cells. So that was pretty suggestive that the hub was playing some part in this regeneration of the somatic stem cells. So I've shown you that when we kill these cells, we actually see a loss of quiescence in the hub cells, suggesting that they are going to make new stem cells. So how do we figure this out? We repeated this experiment again, but now we introduced a transgene into the flies that would mark the hub. And it's going to mark it temporarily. It's not going to mark the cells indefinitely, but it should mark them for several days. So this is the marker we used. This is just the testis again, and you can see the hub here. Now it's very, very green because we've actually, the transgene expresses green fluorescent protein. And I have another stain on there that shows you somatic stem cell nuclei and also differentiating somatic cell nuclei. And after we do the ablation, of course, all these pink nuclei are gone because we've killed them. So these are actually germ cells left behind, and the hub is still a nice compact little ball, and the green is all in the hub. But we got very, very excited and all had to take a turn looking at this down the microscope when Phyllis found this, because we had never seen anything like this before. Now you can see this is just 3 days after we let the tissue try to recover from this grievous injury we've done. So the hub is looking a lot less compact, and you can clearly see that there are cells that express both the GFP marker and are upregulating this somatic stem cell lineage marker at the same time. So that was very exciting. And as we expected, if we wait a little bit longer, the hub cells, we know they stop dividing after a week, and after 2 weeks, everything is returned back to normal, and this marker has become restricted to the hub again. So we thought that was very nice evidence that the cells in the hub were actually starting to leave and start to express stem cell markers and then go on to become stem cells. So from that, we conclude that these hub cells are actually capable of becoming stem cells. So, this is what I showed you. And we're hypothesizing that these hub cells are the source of the new stem cells after we ablate the originals. The other thing we noticed that was really striking is that in these testes that recovered, I showed you a picture where there was only one hub, and every testis only has one niche normally, which is kind of convenient. And we would start to see things like this, where this is 3 whole new stem cell niches that have shown up in this tissue. And the one we think is the original one up at the very tip is not even in this point of view. It's in a different section. And so we got very excited when we saw this, because not very much is understood about how you control the number of niches that you get in a tissue. But each one of these has its own little set of somatic stem cells and a set of germline stem cells. And we know from other experiments that not only do they look like niches, the cells are active in there and they're dividing. And it turns out that there's a reason there's only one normally. The tissue does not do very well with 8 niches, which is what this one has. The cells sort of get all jammed up and it stops functioning. So more is not better as far as stem cell niches. But we're very excited about this because now we have a system to study genetically how you control the number of stem cell niches you might get in a tissue, which is really cool. So, I'm going to sum. I've shown you that when you ablate somatic stem cells, it causes the niche to actually acquire properties we never knew it had. The hub cells exit quiescence temporarily. They refill the niche. We think that the germline, since it was there all along, is just kind of refilling the missing germline, but that was completely unexpected, that a quiescent cell would become the source of a stem cell. And the other important thing is that this way of replacing missing stem cells is not all that efficient, because it's accompanied by this very dramatic side effect of making too many niches. And I have to say that we, of course, don't know how it makes all these extra niches, and it's probably going to take us a while to figure it out, but it's a very exciting problem for us to work on. And I want to also add that this is a response that we see to this kind of a damage situation. It's not something that we've been able to see happening with normal aging. So we don't think that the quiescent niche cells are normally going to make stem cells. We think it's something that you really have to have a terrific amount of damage to the tissue to promote. And